session. Our moderator is Dr. Angana, and the repertoire being Lakshmi. Our first speaker of the day, Professor Pankaj Seth from National Brain Research Center, Manesar, is with us. Yeah. So I would now request the anchors to introduce our speaker so that we can start with the scientific deliberations. Thank you.
Good morning, fellow learners. As we commence the fourth day of our 5-day workshop on the topic neurobehavioral studies using vertebrate and invertebrate laboratory model organisms, let us harness the momentum we've gained and channel it into meaningful progress and growth. We now begin with today's first session with our esteemed speakers of the day. The moderator for this academic session is Dr. Angana Saikia, Assistant Professor AINN, and rapporteur is Ms. Lakshmi, MSc second year student AINN. Without standing in the way of you and knowledge that is to come, let us introduce the first speaker of the day. Professor Pankaj Seth, Senior Professor and Scientist, National Brain Research Center, is a leading molecular neuroscientist and neurovirologist. His major research focus is on understanding the cellular and molecular basis of neuro-AIDS, neuro-COVID, and Zika virus-induced microcephaly. He has also developed a primary cell culture model of human neural stem cells to study virus-induced neurodegeneration. More than 2 decades of neuroscience research has garnered him multiple accolades that it might take me the entire time to note them down, but the prevalent ones are a Sustained Superior Performance Award by Henry M. Jackson Foundation for Advancement of Military Medicine, US, Special Acts and Service Award by National Institute of Neurological Disorders and Stroke, as well as Directors NIH Merit Award by National Institute of Health, USA. With more than 100 publications and around 3,500 citations, serves standing as a foremost expert in his field, affirming his deep knowledge and significant scholarly impacts. Your expertise is a valuable asset to our learning journey, sir, and we are thrilled to have you here insights. The title for his talk is Isolation and Characterization of Human Brain Cells for the Use of Study on How Viruses Affect the Brain Functions. So let's welcome, sir.
So thank you for the kind introduction. For those who have heard my talk before, is there anybody who has heard my talk before? Nobody? And the reason why I'm asking you this question is because I will go over some of the things which we have done. Mostly since it is a workshop, I am not going to show too much of data. Rather, I will give you summary of what I've done. And more importantly, in the first 50% of my talk would be invested, the time would be invested to tell you how we have developed a model system, importance of a model system, and so on. So if I can have my slides now, here slides. Yeah. So at the outset, I would like to thank MIT AINN for inviting me, for giving me giving this talk, and in particular, Professor Malik. And whenever he says something, we can't say no. And this was anyway a pleasure. I was traveling a little bit, so I had requested him to place my talk today. I'm really thankful for adjusting to that schedule. So as it has been said, I will be talking to you about a model system. And the reason for this is the uniqueness of this model system is that it is a primary cell culture. So if you know cell culture, how many of you, let's make it interactive. It's a workshop, right? How many of you know cell culture? None of you? 1, 2, 3, 4, 5, 6. Okay, maybe good percentage of people. So cell cultures are done in 2 ways, right? There's a primary cell culture and there's a cell line. Have you heard the term cell line? So cell line are usually also derived from some tissues, okay? So when you take a cancerous tissue and if you develop a cell line, so that itself has a capability of rapidly dividing, okay? So it continues to proliferate because it is a cancer in nature, okay? What is cancer? Cancer is simply a uncontrolled cell division process, right? Usually cells divide and then they stop. But in cancer, they continue to divide because that control or the programming of control of the cell division is lost. Now, if I want to develop a cell culture system and I want that cell culture to grow forever, so cancers. One way is that I derive it from a cancerous tumor, then it itself is there. But if I'm deriving it from, let's say, brain of a normal individual, then so I have to make some changes in that so that it continues to divide. Now, why do I want to make it for continuously divide? So that every time I don't have to go back and look for a brain tissue. So we are deriving these human neural stem cells from fetal brain. Now, where do I get the fetal brain from? They're elective abortions, and then the tissues are a waste. So here, these fetal tissues are non-cancerous. So I derive them from the brain, and then I do something to them to make them cancerous. I can do something to make them cancerous. Now, if I do not make them cancerous, then it is considered as a better cell culture system. And that cell culture system is called as a primary cell culture system. So if somebody asks you what are the difference between the primary cell and a cell line, the primary cells, if you make them cancerous or you alter the genes so they lose the control of cell division, in that case, they become cell lines. Okay, is that clear? So cell lines are cells which can continuously divide, but you have to do something to the normal tissue to make them cell line. We will not go into how you can make cell lines today, but I will talk to you about a primary cell culture which we have developed. Now, the advantage of primary cell culture system is that they're as close as possible to the normal tissue. If I am culturing these primary cultures in my primary dish, they're as good as I am studying my brain or the brain of that from the source for which I have taken. The disadvantage of primary cultures are that they start dividing, and after some time, like we all are born, we grow, and then we age and we die. So similarly, the primary cell cultures also follow that pattern. The second disadvantage is that you have to again go back to another fetal tissue, right? And then grow another one. So you have to constantly depend on the source of the tissue. But the advantages are far more greater as compared to the disadvantages. And that is why primary cell cultures are said to be more superior for the studies as controlled as compared to the cell lines. So you understood what is primary cell, what is secondary cell line. So today I'm going to talk to you about a primary cell culture model. And we have developed neural stem cells. The moment you hear the term stem cells, you will think it is to be used for regenerative treatment, because stem cells have the inherent property to differentiate into any cell type of your body if it is a totipotent cell. So like there is a hierarchy in stem cells also. Have you heard about stem cells, right? Most of you would. So they're totipotent, total potential. Pluripotent, they can make all cells other than the placenta. And then multipotent, and so on. So these, what we are deriving are multipotent cells because we are deriving them from brain. So they have already been decided that they will go into brain cell types. All right. Is this clear? Are we all the same page till now? So the hierarchy is just like, vice chancellor, principal, head of the department, class teacher, or department student, faculty. So our lab, my lab at National Brain Research Center, is having 3 research questions. Overall, we are trying to understand how viruses, if they get into our brain, how do they alter the brain functions? Now, the first question which should come to your mind, do viruses get into the brain? And if they do, we are trying to understand what do they do, okay? So we are trying to understand 3 viruses, HIV, Zika, and SARS-CoV-2. Now, why these 3 viruses? Because these 3 viruses have been reported in the brain. And individually, these viruses, such as HIV, causes HIV-associated neurocognitive disorder. Mostly, whenever you hear a term, it itself will tell you about it. you can easily, in that blue box, you can make out what this disease will be. It is associated with HIV, so it is called HIV associated. Neurocognitive means it has to do something with the brain, and there also about the cognitive function, and it causes disorder there. It is briefly called as HAND, HAND. Okay. Zika virus, on the other hand, is known to cause microcephaly. Break this word into two. Micro is what? Small. Cephaly is head. So it is a small head size of an infant is Zika virus induced microcephaly. SARS-CoV-2, I'm sure if not you, somebody in your family or friends have gone through this process, this disease of COVID. COVID is caused by SARS-CoV-2. SARS-CoV-2 in long COVID patients, usually they have some neurocognitive discomfort or disorder, which is called as brain fog. That's a very loose term, but it is again defining you. Something like in the fog in winters, you can't see through, right? So similarly, you have a fogging where your brain does not function at optimal conditions. And we have seen that that is very common in long COVID patients. Around 40 to 50% of these long COVID patients have this problem, which is still persisting. And that is now, or the COVID is gone, but the morbidity of COVID continues. So today I'm just going to talk to you about the first two viruses because in the interest of time, and let's move forward to the model system. Now, whenever you want to start studying anything, disease or non-disease, you need a model system. You can, if you're a clinician, maybe you're working on with patients, you may try to understand what are the symptoms of any disorder. Or if you don't want to study disorder, you just want to see, you just want to understand how we see, how we hear, how we speak, how we walk. That also you need some model system. Okay. Similarly, when we started to work with HIV way back in 2004, we wanted a model system. Now, unfortunately, with HIV, uh, there are no animal models for that because this virus does not multiply in animals. It's a human specific virus. That is why it is called as human immunodeficiency virus. If you take that virus, put it in any animals, nothing would happen. You accidentally poke your fingers or introduce it into blood, there's a possibility you may develop AIDS in 5 to 10 years. So it's a human specific virus. So you needed a human model system. You can't directly work with humans. So we started to look for a model system. And that is why we started developing this human fetal brain derived neural stem cells. So this was a need. We did not develop a model system without a need. There was a need to have a human. And since we wanted to study HAND, which is HIV associated neurocognitive disorder, you wanted to have brain cells. Now I had a choice that I develop three cell types. I take astrocytes, I take neurons and oligodendrocytes and all the other brain cells. Or I could make neural stem cells, one cell type, and then differentiate from there. So that was easier. So we started developing human fetal brain derived neural stem cells. Correct? Now it has a procedure which is listed over here, and we have published it in several articles. So I'm not going to go into that technical details, but to tell you the source of our tissue was always 8 to 16 weeks or the first trimester of the pregnancy. The pregnancy is of 9 months. It has 3 trimesters, 3 months, 3 months, 3 months. We try to focus in the first 8 to 16 weeks, which is falls under the first trimester. Why first trimester? Because that is the time when you have the maximum number of neural stem cells and the brain is developing in uterus. And it is continuously having a pool of neural stem cells, which are making astrocytes, neurons, and all the brain cell type. And the brain grows, the head grows in the body. Okay. So we looked for aborted fetuses between 8 to 16 weeks. Up to 20 weeks, it is legal. After that, only under when the mother is under threat, the abortion are allowed. But 8 to 20 weeks, abortions are allowed. So we looked for that age group. We did a lot of culture. We passaged them. If you do cell culture, every time you divide the cells and change it, the flask or the that is called as passage. And then based on the morphology and cell specific markers, we made sure that the cells what we have are actually neural stem cells. If I have a mixed culture, that's not a good thing. So if I want to, let's say if I want to grow roses in my garden, and I want only roses, so I don't want at that time any other flowers. So exactly, since we wanted to make neural stem cells, we had to ensure that we have a purity of the cell population, correct? Now let's move on and look at this. Now this slide which you're seeing over here is day one from the brain of the fetus, okay? So you take fetal brain tissue and you bring it into small pieces by passing it through pipette, and it's a very soft tissue. It breaks up, okay? We don't use any enzymes because that may harm the cells. You remember, we have to have a primary cells as close to brain. So we just give it an environment which is called as media, which is similar to what perhaps the brain has or whatever we have understood so far. So in that media, we grow these as brain pieces. And at day one, you can see that something is sprouting out. Just imagine this is a sun and sun rays are coming out. Or you put something to grow and it sprouts out. घर में sprouts बनाते हैं ना, तो उस तरह से समझ लें. And then this is part of brain, and this is the same picture taken on different days up to day 8. Now, if you see this from here, is the same brain part. Okay, but you will notice that the diameter of this is more as compared to this. It is darker also. So what is this is telling you that it is growing, it is multiplying. And this is a region which is rich in neural stem cells. Okay, to start with, we don't take the whole brain. We go to the portion where we know there are a lot of neural stem cells. We pick up, break that part, and culture this. And if you move day-wise, I'm going slow, sir, because I will try to complete in 40 minutes. But I'm going slow so that they understand and I have their attention. So you see that it is filling up that plasticware, and you see from here to here, it's all full. Now let me take you to the next slide, which is again the same brain part, but I've just moved the camera a little bit so to give you an idea how far they are sprouting out. Okay, from here up to here, and here, it's almost filled up. Okay, so this is the speed at which they proliferate, and neural stem cells have two major properties. Any stem cell for that matter have two properties. One is to divide rapidly or self-renew themselves. Self-renew themselves, matlab same cell say they will make another cell like them. So every mother's stem cell, when it divides, it makes two cells. One, it goes back as a stem cell. Second, has a potential that it may differentiate into the cells which can. So in this case, two cells, when they're formed, one of them goes into either astrocytic differentiation or neuronal differentiation. These are the two main types, glial cells and neurons in the brain. So they go into that fate, and the one of them self-renews and keeps itself as stem cell. Now, how this fate is decided, it's another story. We'll discuss it some other time. Now, as I said, when I'm culturing this, I want to be very, very sure that I have, yeah, it may show better. Right. So these staining pictures don't look good unless there's good contrast. Okay, it shows even better over here, but it's okay. You can see there's some blue dots, right? And there's some red fiber-like structures. So here we have stained the nucleus with blue. So if I look at this and suppose there are 50 blue dots, that tells you there are 50 cells in this frame. Okay, and the red fibers which you're seeing are the cell process of that, and that is stained by cell-specific marker. In this case, it is nestin. Okay. Now, as I said, when I'm culturing, I want to be sure that it is neural stem cells. I have to depend on the morphology. If I know how a stem cell looks like, I also have to look for cell-specific marker. Okay. So they are unspecialized at that point. What does that mean? They are not astrocyte, not glial cells, or not neurons, or not oligodendrocytes. So it's just something like when a kid is born, you don't know what that person may become. It may become a doctor, an engineer, a scientist, a thief, a politician, anything. Okay. So at this point, they're unspecialized. Now, based on the need of the brain, Some of them become astrocytes, some of them become neurons. Now here, because we are doing everything artificially, we have to provide them the media conditions and chemicals and signaling so that they either become astrocytes or they become neurons. But at this stage, when I want only a pure population of stem cells, I checked for them and I found that most of the blue dots, which mean all the cells, are red. I have stained the cell specific marker nestin with red. I had also looked for glial cell fiber marker, which is GFAP, glial fibrillary acidic protein, and neuronal marker TUJ1, early neuronal marker. When I looked for any green cell, they are not there, which means all my population is only nestin positive cells, which means it's a pure population of neural stem cells. That's one way. When you become scientists, everything you have to prove by multiple ways. So morphology, cell-specific marker, and you have to show the positive marker as well as the negative markers also. And then that's not enough. So you then try to see what are the other properties of neural stem cells. And the other property is to form sphere-like structures, neurospheres, neurospheres, brain spheres. Okay. So they give whatever the properties should be was present in this population. So this gave us more confidence that they are neural stem cells. Now, how do they look under a microscope? This is how the neural stem cells look like. Yes. So this is how neural stem cells look like. When you differentiate them into astrocytes, which means you have to give them a media condition which will turn them into glial cells. This is how they look, flat cells. And at this point, they're looking like spine-like. I have just stained them also, and this is how they look. Now, again, stem cells were nestin positive, so they are all red. When I looked for glial markers here, they were negative. But when I gave them media conditions, all of them turned into astrocytes. This doesn't happen overnight. It takes 21 days to move from stem cell into astrocytes. Similarly, when I gave the media conditions for neurons, all of them converted into neurons. Now, why I'm saying all of them? Because if you see again, the blue dots, most of the blue dots have the green color also. That means all the cells which are there are astrocytes, correct? Now, what are different structures of astrocyte? Why glial cells of this type are called as astrocytes? Aster means star. And if you look at these cells, most of them are star-shaped cells. That is why they're called as astrocytes. Also, please remember, astrocytes are the most abundant cell type of our brain. For one neuron, there are 50 to 100 astrocytes. So you can imagine, we always say we have 100 billion neurons. Imagine how many astrocytes you have. Multiply by 100. Okay. Now they're there for a purpose, and that is they are very important brain cell type. They're more important than neurons. Often everybody worries about neuron, but now the days have come that we have recognized the importance of astrocytes. And they do a lot of things such as energy metabolism, glutamate homeostasis, and support neuron function in many, many ways. And the marker which we have used here is GFAP. Please write it down. It is called as glial fibrillary acidic protein. It's fiber-like structure. It's an intermediary fiber. And if you look at all these pictures over here, you will see fiber-like structures in them. Okay. This is what it is stained for. Now, for the other cell type, the neurons on the right side of the previous slide, which you saw, you again have to make sure that they're neurons. How do I make sure that they're neurons? They should be long, branched, but any other cell can also be like that. So we wanted to then check them again with cell specific markers, and we looked for TUJ1, which is beta 3 tubulin, early neuronal marker, which is present over here. Again, look at these three frames of the cells. How many cells are here? Blue dots, 1, 2, 3, 4, 5, 6. And if you come here, all 6 blue dots have some fiber and they are coloring for TUJ1. Now, we also wanted to show neurons are of various types. We wanted to look for whether they are dopaminergic or some other cell type. For dopaminergic neurons, you want to make sure that they are TH positive, which is a precursor for that, which is called as tyrosine hydroxylase. I'm not trying to be technical because I lose my audience. I want your interest on this to understand how any cell type which you grow, you have to confirm with cell specific markers. So we are trying to use different markers to make sure that what we have and what type of neurons we have. This is shown here in all three cell type in different cultures we have tried to see, and most of them are TH positive, which means at the end, the neurons which I have made, primary cultures of neurons which I have made from human brain are actually dopaminergic neurons. Okay. And if you want to study Parkinson's, this is the best model for you. Now, as I said, we had made astrocytes, we had made neurons, but we were not sure whether they can also make oligodendrocytes. Do you know what oligodendrocytes do? Oligodendrocytes provide a myelin sheath on the neuron axons mostly. Okay, just like you have these electrical wires going around, you have an insulation over that. So that myelin sheath is provided by cell type called as oligodendrocytes. It is not a continuous insulation. It is broken at parts because so that the nerve impulse can go fast. So this is a unstained picture of oligodendrocytes. Again, we had the neural stem cells. We gave them different media to make oligodendrocytes. When we looked at the cell markers for oligodendrocytes, we found that they are not nestin positive now. They are mostly green or orange, which is O4. It is a marker for oligodendrocytes. And blue is DAPI over here. Okay, so all the cell types which we have this green marker, which is confirming that they are oligodendrocytes. Now, as I said, we can culture them as adherent culture, and they can also make sphere-like structures, which are called as neurosphere. So these neurospheres were formed if you don't grow them with adhered to plastic. Okay, you have to coat the plastic to adhere them. Then they grow as sphere-like structures. Now, is this sphere a collection of cells, which is just, you have 1 million cells and they have all come and made a sphere-like structure or not? So how do you find that? So you have to follow, you put one cell in culture, put the microscope and come every day and see what is happening. If you put one cell and tomorrow you see 10 cells, that means that one cell is given growth to the 10 cell, which is possible because neural stem cells are supposed to self-renew themselves. So to confirm that, we use time-lapse microscopy. टाइम लैप्स माइक्रोस्कोपी आपने कई बार देखा होगा ना, वो क्लाउड्स चल रहे हैं और सन ऊपर आ रहा है, उसमें क्या होता है, एक कैमरा होता है, it takes pictures after you can program it and then you make a movie. Something similar what we do at here. Now, using this cell culture model system, we have published several papers from the lab as well as in collaboration. And these papers mentioned in blue have got special attention from the journals as well as from Nature India, which basically just tells you that the model system is accepted worldwide. Now, is this model system easy to make? No. Is it useful? Yes. How many labs in the country make them? One. How many labs in the world make them? Maybe a dozen. So why is it so difficult? It is difficult for two reasons. One, it is very difficult to get aborted fetuses in time, and then the technology and the expertise it is requires. And two, because it is very, very expensive, because the stem cell research is as such expensive. And then usually people go for low hanging fruits. They prefer cell lines, less jhanjat, easy to culture, and kuch jaldi kharab nahi hota hai. But these are very, very difficult. This is like growing babies, basically. Because if you go wrong, it is just like snake and ladder game. At the final experiment, if your culture goes wrong, you're back to square one. So now, as I said, how are those spheres formed? If you use time-lapse microscopy, and if this video works, you will see how one cell divides into two, into 4, 8, 16, and then I lose the count. But in 48 to 72 hours, you will have the sphere-like structure. Now that confirms that the sphere you have is actually derived from one cell, right? And that is again another advantage. And the other beauty of this is that all of them are similar. and they're all stem cells, because if they were not stem cells, their morphology would have changed. Now, here is an outlier. You see? So what has happened to this cell is basically it has died or has about to start differentiation. And that is why there's a hollow space over here. Okay, let's not get into detail. I just wanted your attention to this particular type, but most of the other cells look alike. Now, if they are stem cells, again, they should give rise to neurons and astrocytes, right? So you want to confirm that, and that we will do in another slide over here. So here's a neurosphere. Again, you stain for all the possible markers of stem cells, nucleus, astrocytes, neurons. Here we just look for neurons and stem cells. And what we find is here is a new neuron, because neurons, we have stained them with green. Okay. Now, if the major problem of neurological disorders is damage to the neurons, so neurons die. The other problem is neurons don't divide. Once they are formed, nature, they're terminally differentiated cells. Unlike other cell types of our body, skin cells, astrocytes, oligodendrocytes, neurons do not divide. We are formed with some numbers of neurons, and then we are stuck with that number over the period of years. And actually, that neuronal number goes down. Recent research in last 10 years have shown there's little bit of adult neurogenesis. In adult also, some stem cells are there, and they give rise to neurogenesis. Neuro, neuron, and genesis is making of new neurons. So neurogenesis occurs, but that is limited and does not compensate for the neurons we lose over the period of years. Okay, but there's adult neurogenesis. This is the new concept, not new, but last decade has shown. Now, the whole process, if we could grow enough neurons in our brains, if we could induce the stem cells of our brain to make new neurons, most of the problem of brain disorders may be controlled, or we may have a better outcome. And that is what everybody is trying to do, is make new neurons, either in culture or in brain or in animal models. So here you're seeing one new neuron being formed. Okay. Now, if I zoom into and take it another picture over here, and you will see now cells are green or yellow and orange. You don't see any red cell. Red for nestin, which is neural stem cells. So the green cells is showing that the neurons have been formed. Yellow and orange is showing that they are trying to lose their red stain and start taking the green stain. That is why there's a merge picture is showing as this. So they are under differentiation. They're not full neurons yet, but they're moving towards that path. Like you guys are moving to become a scientist or a researcher or whatever. So you're in between. That's exactly what they are. Now, once we made astrocytes and neurons, again, let's go back to the real situation. Why are we doing all this? Don't forget, we want to understand what HIV does to the brain. Now, HIV, when it is there in the brain, it can affect all the cell types, right? So we want to study, and in real brain, as I said, for every neuron, you have more astrocytes, and they're all living together. There's no one region in the brain which you have only one type of cell type. Okay. So we want to, again, have a co-culture system, because you want astrocytes and neurons to grow together. So once you have a pure population of astrocytes and neurons, then you want to co-culture them. Now, somebody can ask, why did I make these separate cell types? Because if I want to study the effect of something on astrocytes alone, I can't do that in mixed brain culture. So based on the research question that you have, your model system will be made. So to have all the possibilities, I started with stem cell, and then I can do separate cell type. I can do mixed cultures. I can do everything with that. That is why we took this approach. Later, we made a co-culture system. All this is published. I'll be happy to talk to any one of you today or later if you want to understand more. Now, here's a co-culture. Now, look at this green cell. Again, it's a neuron. And all the red cell here are not this time nestin positive, not uh, neural stem cell, but they are astrocytes. Astrocytes have the glial fibrillary acidic protein. GFAP is the marker for astrocytes. And you see, I have purposely plated more astrocytes as compared to the neurons. So what I do, I plate 100 astrocytes on the in a cover slip. And then after, when they have attached, I put just 10 neurons on them. Okay, this is just a ratio I'm telling you. So I have plated that and then I let it grow. And what you see is amazingly that this neurons tries to grow its processes and looks for an astrocyte. And the moment it finds an astrocyte, it stops there. Now, why is it trying to reach out to an astrocyte? Because it depends on the neurons depends on the astrocyte. Like I said, neurons get everything what they need from the astrocytes, which is glucose, growth factors, and other things. And what they do not need is excess of neurotransmitters. They make some neurotransmitters, they leave it at the synapse, and then there's sometimes excess of that um, neurotransmitter, which is glutamate in particular. That has to be cleared out by somebody. So that job also astrocytes do. So they take away the toxins and waste material of the neurons and they supply the goodies to the neurons. And that is why there is a constant crosstalk between astrocytes and neurons, which is established in this co-culture system. Now, how do I know that this co-culture system is actually active and neurons are dependent on astrocytes? Now, if I put any toxin, now here I have tried to put a viral toxin, which is called as HIV TAT protein. Now, HIV virus, now we are coming to the subject now. The HIV makes, all viruses have proteins for themselves, for their multiplication, for causing toxicity, for whatever function virus wants to do. One such protein is TAT. Okay. This TAT protein is actually very neurotoxic. Neurotoxic. It is bad or it's toxic to the neurons. So if I take recombinant protein of virus HIV TAT, I put it in the co-culture system. The first thing happens within few hours is that neuron which has made that extra effort to go and touch the astrocytes retracts it from themselves because now the friends of the neurons have turned foe. We have a review article called Friends Turn Foe in HIV Neuro AIDS. Read that. It's a very well-written review. So when do friends turn foe? When there's a problem. अपने हम लोग ने भी देखते हैं ना आपका कोई प्रॉब्लम आ जाए तो सारे फ्रेंड्स गायब हो जाते हैं जब आपके पास सब कुछ ठीक है everybody is around. So similarly, what happens? Neurons start retracting because astrocytes, when they have the toxin, they start making certain chemokines and cytokines, which are not good for neurons. And that is when neurons realizes, you know what, this friend has turned foe. I don't want to be with it, and it retracts. If I wash off the media and if I remove the TAT protein, this neuron goes back and attaches there. Okay, if the conditions are favorable, it goes back. If not, they die. And before the neurons die, they make these bead-like appearance, one bead, second bead, third bead. And if the toxin is left there, neurons eventually die. So this is exactly what is happening in our brain. Once the virus makes its way into the brain, it makes these proteins and neurons start dying. Okay. And that is why you have the neurocognitive disorders in HIV patients. Okay. Now, what is the potential of this neural stem cell model which we have made? It is immense. One can understand, or we have understood a lot of things and provided huge input into the field of neuro AIDS using this model system, where we've used this model system to understand how HIV affects the stemness of neural stem cells. So the advantage what we had of individual cell type, I could put the TAT protein or the virus on the stem cells and found what it does to the stem cell. I can take that virus and protein, put it while they were differentiating into astrocytes or neurons. If I put it at that step, I will see whether the differentiation process is altered or not. If I want to study what happens after differentiation, effect of HIV on astrocytes, effect of HIV on neurons, effect of effect of HIV on neural stem cells, effect of HIV on co-culture, effect of anything for that matter, I can use this model system. And then later on, when the outbreak happened of Zika virus in Brazilian Olympics in 2016, there were a lot of cases reported of microcephaly. Microcephaly, remember the abnormally small head size of an infant when it is born, can happen by many ways. But in Brazilian Olympics in 2016, it was found that there were microcephaly cases suddenly went up. Number of microcephaly cases went up, and they were going up in areas where there were outbreaks of the Zika virus infections. Okay, we'll come to that a little later. So then that is when we use this model system to understand how Zika virus causes microcephaly. And then later on, when the SARS problem came in, we also looked for how, and we are continuing to look for how SARS-CoV-2 and its virus, its proteins are affecting neurogenesis. Now, I told you that HIV goes into the brain, and you simply believed it. You should not, never. If you want to be a researcher, ask questions. A virus which is known to bring down the immune system of people, that is what its primary function is. HIV, human immunodeficiency virus. कोई ये नहीं होता है human brain neurocognitive virus. Its primary function is to bring down, the immune system. But this way into the brain, and the strategy which uses is this one. It uses a Trojan horse hypothesis. This picture tells you how one fort was kind of broken in, is by presenting this horse in which the enemy's soldiers were there. That's exactly the strategy what this virus does. If you look at this cross section of HIV, and if you follow this animation, you will see this HIV virus gets into the blood, and since blood goes to all organs, including brain, it makes its way through the microvasculature, gets into the brain. And if you look at this microvasculature, you will see the cross section of the brain. Okay. Now look at this. This is the endothelial cell lining, which forms the blood-brain barrier. These are your RBC. Here is a small viral particles. If you can see this white particle over here. And here is one blue cell, which is called as monocyte. So this is the horse. This is the enemy. And this is the fort. This fort does not allow this teeny mini viral particle to pass through, but somehow it allows this monocyte to pass through. So this is a selectively permeable barrier, which does not allow things which are not needed in the brain. Virus is not needed in the brain. So there's a natural mechanism to stop that. However, this same selectively permeable barrier allows this monocyte. Why it allows this monocyte? Because brain does not have its own immune system. So the monocytes go and do a surveillance. And if there's some need, they will turn into a macrophage and they will become an immune cell. So this is similar to what happened today when I walked into Amity, or I was driven into Amity. So there was a barrier, considered as a blood-brain barrier. There was a guard who looked for whether we should allow this. So that was this blue cell, and I was the viral particles. They did not check me through, and they allowed me because I was coming in the car. If I was coming alone, they would have stopped me. So I'm just giving these similes so this remains as a permanent memory to you. And that's exactly what happens. And if I continue this animation, you will see that this is a virus loaded monocyte. Viruses cannot pass, but because monocytes can pass through the barrier, this infected monocytes gets access into the brain. And the blood-brain barrier accidentally allows this monocyte to come, and then the viruses are spread. And the spike protein of the virus, actually not the spike, but GP120 docks to the receptors on the cell, and you just saw infection of a brain cell happening after crossing the barrier. What does it do? It causes damage, dementia-like symptoms. Again, as I promised you, I will not show you data, but give you slides which will help you understand how one should do research. Neurons are not infected, but they are affected. So they die because the viral proteins and the virus. This is called as direct and indirect cell death. And most of the death actually occurs because of infection of the glial cells. Glial cells are astrocytes here, and astrocytes, as I showed you, they're very important. If astrocytes become sick, the neurons are eventually affected. Now, astrocytes, I give a simile of lady of the house. You have a maid, you have mom, you have somebody who's looking after the house. And the person is healthy, happy, everything is fine. You don't even realize how much work that person does. Person falls sick. First day, some problem. Second day, more problem. Third day, more problem. And then eventually the whole system goes down. That's exactly how astrocytes support the brain function and neurons. So once astrocytes are sick because of infection of this virus, and astrocytes are infected by HIV, the problem starts. And eventually what happens is death of these neurons. So I will quickly now go into all the 4 research questions we asked using this model system. One was whether the stem cells are affected by HIV. Very quickly, we just took again neural stem cells. We looked for cyclin D1, which is a cell cycle regulator. So when every time the cell has to divide, it has to go through 4 steps. Okay, there are 4 stages. And CDKs are, cyclins are these stages which push the cell into the next step. What we found was when you have TAT protein in the system, these steps are interrupted. The cyclin D1 goes down and it does not push the cell, so it gets stuck in one phase. That's exactly what. So you see these green cells, though these are not neurons, these are not their nestin. Here, nestin is not stained. Don't get confused by the green stain only. Every time you can tag any antibody with these 3 colors and more colors actually. Here we have stuck it with cyclin D1, and you see a lot of cyclin D1 present in normal neural stem cells, but when you come with TAT protein, the all cyclin D1 is gone. Cells are there, but cyclin D1 is gone. If you look at another red stain here, which is Ki67, which is a marker for cell division. Ki67, you see how many red cells are there or pink cells are there in this one, and you look at very few pink cells are there, which means the cell division rate is also gone down. And that means that the cell division of neural stem cells is affected when you have viral protein. Now, later on, we went into mechanisms of each of these steps, and we figured out how TAT protein is bringing down the differentiation. And this is the person who did this work. She's now currently a postdoc. So this is the paper which got actually attention of Nature journal also. So then Meher went out and looked at how astrocytes are affecting the neurons. So she figured out that how astrocytes are causing toxicity to neurons. Again, I'm not going into details, just one slide each of each research question we asked using this model system. And here, Manju asked this question to understand how, again, astrocytes are mediating neuronal death, because she worked out the entire pathway receptors by which astrocytes are causing the death. This paper was also highlighted on the cover page of the journal. Later on, Chitra further dissected out the process. And this is how you understand a disease. How is neuropathogenesis occurring is studied in these different studies, okay, research questions. Here we use the co-culture system. Earlier we used only stem cells and then co-culture systems. Then coming very quickly to Zika virus, similarly, just in few slides, I'll show you. Time is over. Okay, I'll just breeze through it now. So this is microcephaly. This is normal head size. Typical head size should be this dotted line, but microcephaly is happening. So if you remember, I told you that the entire growth of the head occurs in the first trimester. And it is known that if the Zika virus infection occurs in a mother in the first 3 months of her pregnancy, there's a possibility to 7 to 8% or up to 20% infant born to such mothers can be microcephalic. But if the same Zika virus infects a mother which is infected in the later part of her pregnancy, nothing happens, no problem of microcephaly, because that also tells you that the development of the head actually occurs in the first 3 months. And that is why if you know somebody in your family, you will see that they go and do ultrasound during pregnancy 3 times. They measure the development of the child, not for Zika virus, but for the overall development of the child. Okay, and if there's any anomaly, there's advice to abort the child. Okay. Now, so this is microcephaly. Is it real? Yes. Here are some pictures of microcephalic child. They have very poor outcome in life. Many of them die as stillbirth at the final stages of their pregnancy, or just after they're born, they die, or they live maximum up to 12 years. And this is, so it's a very bad disease that way. Okay. Now, how is it spread? It is spread through the same mosquito which causes malaria, dengue, chikungunya. So if you have a mosquito bite, be aware that may also be carrying Zika virus. And Zika virus is there in India. It's not a problem of forest only. It has been reported. Several outbreaks have happened in last 7 years. Every alternate or 2 years later, you have this problem of Zika virus outbreak. Fortunately, we have not reported any Zika virus-induced microcephaly in India because it is a different strain, and so far it is not. But does that mean it may not happen in India? No, it can happen in India. So it's better to be prepared And this is what Zika virus does. So it goes into the mother in the first trimester, and it causes microcephaly. And we are trying to understand how microcephaly occurs. So what do? You try to take the virus, you try to study the development of the child. Okay. Now I cannot grow babies on the lab, right? So we try to develop head in the lab. Now I can't even develop the head in the lab. So I see how head is developed. Head is developed by neural stem cells. So I take the neural stem cells, I throw the virus or the viral proteins, and I try to understand, does it affect the proliferation rate? So we found that it affects the proliferation rate, and this is how it does. Okay. These are various mechanisms. It affects the microRNAs and notch pathways, and the sphere does not develop. Okay. So that sphere which you saw in animation, if you have the viral protein, it does not develop. With that, I will just wind it up and just show you the final slide, that we have several lines of neural stem cells in the lab. We share it with other researchers, and we have standardized methods by which we can make different type of the brain in this lab. And the advantage of this is that you can study at various stages of neural stem cells. With that, I thank you all for your attention, and I'm sorry I stepped over the time. I'll be happy.
Good morning, sir. My question is that what is the basic lifetime of one culture which you extracted, and how often do you need these to take it?
Very good question. Since it's a primary culture, it's a reasonable question to understand how long we can grow. So we can grow them for up to 16 passages. 16 times we can expand them, which gives us enough window to do all these experiments. And once they start getting slow, we differentiate them into astrocytes and neurons. So we have a good window. And from one brain sample, we can make millions of these cells because they grow very fast at the early stage. So we cryopreserve them. And that has been very helpful. Usually primary cells, you cannot cryopreserve them. Cryopreserve is basically putting them in liquid nitrogen. And we have been successful in bringing them out and culturing them again.
Thank you, sir, for providing us with valuable insight. Now I request our moderator, Dr. Angana Saikia, to facilitate our speaker with a moment. We are profoundly grateful to you for, yes.
Yes, you know, doctor. Yes, I do. I am meeting that. So please input what you have a pen drive or something to put in.
It's better because they record everything and somebody the centrally and everything, so you can. Do you have the pen drive or something? Okay, so till Dr. Mona speaks me because I know his function. Krishna, Dr. Krishna, he has joined the university. I will now get another flavor. This will be in Drosophila. But I don't know much about Drosophila. But then if you have any questions, then let me Dr. Pankaj Sage, I think you can continue to be set up. I hope you will not mind. Yeah. Yeah, please go ahead. No. Sunai, everybody has to listen, not that. Another mic. They record everything. Yes, go ahead.
Hello, sir. My name is Mehak. I'm from BHU. So my work is actually on assessing the toxicological effect on the brain cells. So I am also performing primary culture. So I have this query that we are doing mixed cultures. So we yet don't know on which specific cell it affects. So do we have to do the mixed culture and then separate it and see it individually on every cell, or we can co-culture it and
So
form
if
an
you are
hypothesis?
looking at just toxicity on neurons, you should ideally do separately first. Okay. But again, as I said, neurons don't grow by themselves. They grow with astrocytes. So mixed brain culture would be better because your toxicity of your compound may differ in the presence or absence of the neighboring cells. So I would suggest go with mixed brain culture. But if you want to specifically find out what pathways in which cell type is altering because of your toxin or your test agent, then you have to do separate cell culture.
Thank you, sir.
Good morning, sir. I'm Rishika from Central University of Punjab. So I'm working on bone marrow derived mesenchymal stem cells. So I'm facing a problem that the cells are not getting trypsinized. So what could be the possible reason according to you for that?
So trypsinize from the
Not getting detached from the T75 flasks. In
okay,
which
so
I'm
one of the thing could be that what we always go wrong is we don't pre-warm the trypsin. So trypsin is an enzyme. Are you pre-warming it at 37?
Yes, I've tried so many things like pre-warming trypsin, also changing the flask. Everything
Okay.
I've tried.
So I would tell you try this if you have not done this. Take your media and remove the media, of course. Take your flask, remove the media, wash it with PBS once. So trypsin gets diluted if there's any BSA or protein left from the previous media. Okay. That is why when you're doing trypsin at the end, once the cells detach, you neutralize the trypsin. What is happening is first check the percentage of the trypsin what you have. Use that trypsin to trypsinize some other cell type and see if it's working. Is it working on other
Yes,
cell?
sir, it's working.
So the percentage is fine. Now wash it with wash your BSA off or the whatever protein is there in your previous media. Remove all that. Put warm trypsin because trypsin is an enzyme and it works optimally at 37 degrees. And then when you add trypsin, you add enough trypsin and then tap the cells flask also. Sometimes that is also required because if you leave the cell with too much of trypsin, that will also kill your cells. So try these three things and write to me. I will try to figure out what is happening.
Thank you, sir.
Yeah. Yes. Yes. For 1 minute. Keep watching it because the moment they become round, she's right. Keep it at 37.
Thank you so much.
Thanks a lot. Okay, so now we can start.
We welcome you, sir. We're extremely privileged for us. So, following a comprehensive discussion with Professor Seth, we are poised to welcome our next speaker to the platform, Dr. Krishna Melnatur, Assistant Professor in Biology and Psychology, is an eminent researcher at Ashoka University. His research dwells into various aspects of Drosophila biology, shedding light on sleep regulation, learning mechanisms, neural circuitry, and gene expression dynamics. Through innovative methodologies like the sleep nullifying apparatus and investigation into the role of sleep in special learning, he uncovers fundamental insights into the intricate workings of the Drosophila brain. So these findings contribute significantly to our understanding of sleep physiology, cognitive process, and neural development in this model organism. Offering valuable implications for broader neuroscience research, he has received multiple awards, few of which are DBT Ramalingaswamy Re-Entry Fellowship 2021 to 2026 and SERB Co-Research Grant 2023 to 2026. His multiple publications and book chapters has solidified his reputation as a leading authority in his field, demonstrating his profound expertise and scholarly contributions. We're delighted to have you with us and look forward to the enriching exchange of scientific ideas and knowledge that you will inspire. So the topic of his session is Sleep is Plastic and Supports Plasticity. We welcome you.
Thank you so much. Hello. Hello, everybody. Thank you. Thank you very much to Professor Malik for inviting me. It's a real pleasure to be here. And thank you, Spriha, for that very generous introduction. So as Spriha mentioned, my name is Krishna Melnatur. I started at Ashoka University about 2 years ago, a little over 2 years ago now. And so we use flies as a model and Drosophila as a model, primarily Drosophila. We're thinking about other things as well, but primarily Drosophila as a model to understand really fundamental questions about sleep, sleep regulation and sleep function. So the work I tell you about today is mostly work done before I got to Ashoka. Okay, and since this seems to be a lot about techniques and so on, I thought I would try to emphasize a little more about the kinds of techniques we use. Maybe it's a little bit different from probably what you've heard so far, because flies have a real wealth of genetic tools and circuit tools and so on. So we can get at really very precise kinds of manipulations and at really high resolution analysis of how sleep can change circuits and so on. Okay, and it's also maybe a little bit different from what you've heard so far, because some of it is a little bit inspired by ecology, which maybe you have not heard so much about so far. Okay, but I think it's still a different perspective, and I think an important one. So my talk is titled "Sleep is Plastic and Supports Plasticity," and so the idea really is that sleep itself can be plastic, that it's modifiable both in response to kind of environmental changes that an individual animal might encounter during its life, but it also can in turn support plasticity in learning. So what I thought I would start with was, the first part, which is sleep plasticity. Since something that maybe not everybody is very familiar with, but and so this is a picture that has really changed in the last 20 years or so. So, I'm sure you all know Professor Malik has done really wonderful classic work on sleep and mammalian models. But in the last 20 years or so, it's become fairly clear that sleep is essentially near universal. So it's been found in pretty much all organisms that have a nervous system. This is from a review we just wrote recently. And so something that stands out a little bit when you look at this is that there's great diversity in the sleep behavior observed across these different species. And this diversity is not a bug or it's not a glitch, but really it's something that reflects how sleep evolved. The sense that sleep evolved to be plastic. Okay, so it evolved to be modifiable. It's modifiable, both, like I said, both in terms of individual experiences that animals might encounter, for example, by learning and so on, but also kind of ecological niches that entire species might inhabit. Okay, and if you want to understand the neural control of sleep, understanding something about this plasticity is important as well. So I thought I would give you some examples of this sleep plasticity. And so here's one that most people are probably familiar with, which is starvation. And this has been seen, in pretty much all animals. So this is examples from flies, but this has been seen, of course, in humans as well, right? For example, during Ramzan, which we're in now, right? So, so in these examples here from Alex Keene and Matt Timken, experiment they did was to put flies on regular food. Okay, and we'll come to how we measure the sleep in a minute. But first day, you have flies on regular food. Second day, you starve them. The third day, you bring them back to regular food. And we're monitoring sleep, throughout, right? And what they found was that flies actually, so here's the percentage of time they're sleeping. And what they found that is that on the day that they're starved, which is middle day, they sleep much less. Okay, and that's interesting, but not terribly surprising. But the really interesting thing here was that when you bring them back to regular food, so normally if you sleep deprive an animal, including flies, they will show a homeostatic rebound. That means that they will sleep more or sleep deeper the day after they lose sleep. So, for example, we can mechanically agitate flies and they will lose sleep. Next day, they make up for some of the lost sleep, right? But here, they're not making up for the lost. They've lost sleep. They don't make up for it. Okay, that's interesting. And the second thing that was interesting was that they can still learn. Okay, so normally, so again, if you mechanically sleep deprive flies, they don't learn the next day. But here, they've lost sleep, but they still learn. Okay, so that suggests that starvation is somehow something different. Okay, so it's a different kind of waking state. They're awake, but somehow they're not accruing some of that consequences that you would normally associate with being awake. Okay, and so here are some other examples, a little more ecologically inspired. And so this is work from Niels Rattenberg and colleagues. And so they were looking at these birds, they're called pectoral sandpipers. And these are birds that mate in the Arctic regions. And in the summertime, where it's light basically 24 hours a day. And they have this long mating season. And during this mating season, these birds essentially sleep very, very little. Okay, the male birds sleep very little, almost nothing, and they spend a lot of time fighting. And fighting other males in competition for females and competition for territory and so on. And so what they found was that actually the males that slept the least seemed to sire the most offspring. Okay, so again, this is a little bit surprising because you think sleep is so critical for evolutionary fitness and so on, but somehow they can seem to do without it. Okay? And here's another example. This is from Jerry Siegel. And so what they found here was they were looking at these dolphins, and what they found was that right at the time the dolphins give birth, both the baby dolphin and the mother are essentially continuously active for a couple of weeks. Okay? So there's pretty much no sleep for about 2 weeks. And apparently they do just fine. They have great memories and so on. And this is a time, particularly in early life, where sleep is really critical. Apparently, they can dispense with it altogether and not suffer much for it. Okay? And so it's not just birds and dolphins and so on. So here's some example of a big land mammal, which is elephants. again from Jerry Siegel. So here they found that they're looking at these elephants as they were migrating from one site to another site, and what they found was that these elephants actually don't sleep pretty much during the time of this migration. Okay, they're walking from one place to another place. They forgo sleep altogether for about 2 days. They get to the new place, and again they don't show a rebound, so they don't make up for that lost sleep. Okay. And so here's another example of sleep plasticity. This is more about clocks than sleep per se, but so here they're looking at these rats. They're called Norwegian rats, and so they normally, of course, rats are normally nocturnal. They sleep during the day and they awake at night. But here they were put in a kind of a field, a midden, where they're under predation by a fox, which is also nocturnal. And what they found was that they actually switch their sleep-wake behavior. Now they become diurnal. Okay, so it's a 180-degree change, right? So they modified their sleep behavior to now be awake, right, during the day and to sleep at night. Okay, and again, this might be somewhat surprising because these animals are actually changing their behavior to be asleep at a time when their predators are active. And you might be a little bit surprised by that because you think that you might get eaten when you're asleep and so on. But most animals have a safe place where they go to sleep, including these rats which burrow in this hole in the ground and so on. So it kind of makes sense. So they choose to go to the safe place and ride out the night. So these observations have been put together by some people, including Jerry Siegel, to put forth this idea that sleep evolved to maintain animals in a state of what sometimes called adaptive inactivity. Okay, or we sleep to stay out of harm's way. So most people that think about this think about it just about as the timing of behavior. It means you do behavior that kind of appropriate. Okay, but we think that it might also have some kind of adaptive role. That is, you're in an unexpected situation. You might sleep to help your brains adapt to this new situation, figure out new strategies to survive, and so on. And maybe if there's time, I'll come back to that. A little bit about how we. So we want to see, if there was something to this idea, and I'll tell you a little bit about what we did to get there. Okay. But first, a little bit about how we define, how we measure sleep. So we define sleep using behavioral criteria, and there's 4 criteria. It's a period of quiescence. So animal has been not moving for a period of time. Clearly, this is not sufficient because you could be paralyzed and not moving, right? So the sleep also has to be rapidly reversible. That means when you stimulate the animal, you have to respond. You have to come out of sleep quickly, right? And this distinguishes sleep from being in a coma or being paralyzed or something. Okay. Associated, and this is a really important one, is associated with what's called increased arousal thresholds. That means that you have to be harder to rouse when you're asleep. That means you have to stimulate the animal more, stimulate the animal harder to rouse the animal from sleep. And this is a really important one because it distinguishes sleep from quiet waking, right? So sitting quietly, with your eyes closed, not moving, right? And last is that it's homeostatically regulated. So that means if you deprive the animal of sleep, you should make up for it the next day, okay? And clearly this last one is a little bit of dispute because I just gave you all these examples where it was not homeostatically regulated. Animals lose sleep, but they don't make up for it. Okay? But be that as it may, these are conservative criteria. So these are the criteria that have been used to define sleep in a number of different animals. And these were criteria that were put forth over 100 years ago. Okay, so they're fairly well established. As to how we measure it, we measure sleep using these locomotor activity monitors. Okay, and so we have you have a monitor like this. You put flies individually inside these glass tubes, and they will walk up and down the length of these tubes. And we have an infrared beam in the middle, which you can't really see, but it's kind of in the middle there. And every time it crosses the beam, you get a little blip, so you know the fly is active. Okay, and a lot of the foundational work in the field had shown that over 95% of flies that have been inactive for at least 5 minutes will show a change in what is called arousal threshold. Okay, so that means that we can use that 5 minutes of inactivity as a threshold to define a bout of sleep. Okay, okay. So coming back to our question here of sleeping stay out of harm's way. So one thing I thought was that there's something to this idea, if you disrupt flight can't fly away. Okay, presumably you should see some kind of change in sleep. Okay, and so this is the first time I'm showing you a sleep plot. Looking through this, so this is we have flies on a 12-12 schedule. So first 12 hours is light, second 12 hours is dark, and sleep is here on the y-axis. And so what you see is at least female flies sleep a lot like adult humans. In the sense they sleep mostly at night, the little noontime siesta, and there's more sleep in the beginning of the night than the end of the night. Okay, and what we found was that if you disrupt flight, in this case by cutting wings, but you can also glue the wings together and various other kinds of manipulations. Any way you disrupt flight, you get more sleep. Okay, and this fit somewhat with what we expected. And so we wanted to figure out how this works. Okay? And this is really the power of the fly here, is to try to get a kind of mechanistic insight, right? And so we found that if you disrupt flight, you can increase sleep. So we think that there's some kind of signal from the wings saying, I can't really fly. And so that feeds back to some central circuit in the brain to say, to increase sleep, right? So what's that input? And how do we find that? So a little bit about some genetic techniques. So we use this tool a lot. It's called the GALFO UAS system. And so what we have here is GALFO is a transcriptional activator. It will bind to its target site, which is called UAS, and activate whatever is next to that. And this is a modular system because we can do different things with it. We, for example, combine a given GALFO with different kinds of UAS constructs. We can express GFP, which lets you see the cell. You can knock down or press different genes. You can activate or inactivate neurons by using different channels and so on. Or you can monitor neural activity, using various kinds of fluorescent reporters for neural activity. So this is a nifty little tool. Okay, so we were trying to figure out, what's a signal from the wing, right? And so these are some neurons that I had found were involved in this behavior. They're mostly here. This is a fly nervous system. So this down here is called the nerve cord, and up here is the brain proper. So I found these neurons here in green, which are in the ventral part of the brain, but they're kind of far from the wings. So if you look at a little bit about fly anatomy, here's a fly wing. It's mostly dead tissue, but you have these bristles along the edges that are chemosensory and mechanosensory. Okay, and they send this wing nerve which comes in here into the nerve cord. And so we were trying to figure out, which of these sensory neurons are important. Okay, and so what I did was to find GALFO lines that express in different subsets of these sensory neurons, and then inactivate them and see if we see a change in the behavior, right? And the tool I used here was a potassium channel called KAR 2.1, which is an invert rectifying potassium channel. So it will hyperpolarize the cell and thereby inhibit the cell. And so now we can express this in different kinds of sensory neurons. And we're looking for the change in sleep after you disrupt flight, after you cut the wings, okay? And so all the controls are in blue and all the experimental lines are in green, okay? So what you see here is that, so there's some mechanosensory and different kinds of chemosensory neurons, and what we found was that if we inactivated just two classes of chemosensory neurons that express these variant glutamate receptors called IR52A and IR76B, that if you block these, we can mitigate this increase in sleep that we see following flight disruption. Okay. Okay. So we found our input cells, and so as we suspected, these IR52A neurons are expressed in the wing. So you can see here's the fly wing with some GFP labeled cells here on the margin, and this these neurons project into the fly's ventral nerve cord, but importantly, there's nothing in the brain. Okay. So this suggests that we found the right cells. These are expressed in the wing, and so these are relevant input cells. Okay, great, and that was great. But we're still missing some part because these neurons project into the nerve cord, but something has to take that information from the nerve cord into the brain. So what's that missing part, and how do we find that? And so to do that, I used a tool called TransTango, the genetics are maybe somewhat complicated and maybe too much to get into, so we'll just simplify it. So the idea here is that this is a really nice circuit tracing tool that lets you find all the neurons that are exactly one synapse downstream of the ones that you care about. Okay? So you can in this case, we were trying to find neurons downstream of these wing sensory neurons. So we can label the wing sensory neurons with IR52A, which is in green, and can use this. Okay, so we can label the wing sensory neurons in green, and then all the downstream neurons will get labeled in magenta. Okay. And so when we do this, what we see is that, so now you have your sensory neurons here in green, and then the downstream neurons are in magenta. And so we found that there's magenta signal next to the green signal. That's good. We're hitting the right kinds of cells. And the interesting thing was that from an anatomy perspective, is you can see these tracks that leave the ventral nerve cord and enter the brain in this kind of beautiful expression pattern that looks a bit like deer's antlers, right? And so this suggests that we found the neurons that we were looking for. These are long-range projection neurons. They connect the wing sensory neurons to the brain. Okay? And so that's what we wanted. We wanted something that connects the wing nerve to the brain. So it looks like we found it. Okay, great. So this experiment is very nice and very pretty, but it just tells you that the neurons exist. Okay, it doesn't give you a way to manipulate them. It doesn't give you a way to figure out what's happening to them and so on, right? So to do that, we have to get what's called genetic access to those cells. That means that you have to find a driver or something that's expressed in there. And that, I did by kind of old-fashioned way by looking at hundreds of images, trying to find one that matches that. And so I found one. It's called 31C06. And so you can see here, this is again the expression pattern driven by this enhancer. And you can see that you can see this green signal here in the nerve cord. And you can see these tracks that leave the nerve cord and enter the brain. So this looks like that TransTango pattern that I showed you. Okay, so it looks like we found the right kinds of neurons. Okay, but of course I have to actually show that these are the right kinds of neurons, not enough to simply say that it looks similar. And so to do that, we did a series of experiments. So here we're expressing two different fluorescent proteins. The sensory neurons are now labeled in magenta, and the downstream neurons. And so we find that again these neurons are close by. Okay. Particularly in this wing neuromere. So of course, the fact that they're close by doesn't really matter very much. We have to know whether they make connections, right? And so to do that, we turn to this tool again. It's called GRASP. And so this is a neat little tool. It's called GFP reconstitution across synaptic partners. And so what you do here is you express GFP in the two. You have two halves of GFP. And you express the two halves and the two neurons you think are making contact. Okay. And where they come close together, you reconstitute GFP, you get green signal. Okay. And so that's what you see cartooned here, right? You have two halves of GFP where they make contact, you get green signal. And then we have another reporter here that labels the presynaptic site so that you know that the contact's actually synaptic. Okay? And so that's what you see here. So you see there's this nice P signal everywhere these neurons are close together. Okay. So that suggests that these neurons are indeed making contact. And of course, we looked at these markers for presynaptic sites to make sure that the contact's actually synaptic. Okay. So now you have magenta signal next to the green signal. That's what we wanted. Great. Okay. So, and that was nice, but we wanted to actually refine because we want to figure out exactly which cells within this 31C06 pattern are involved. And so again, I'm going to just introduce you to the power of fly genetics. So here, I told you about the GAL4 UAS, but now we can make that a little bit better by using something called split GAL4. So here you have and where you basically, I don't know if you've heard of the yeast-2 hybrid. It's a way to tell whether two proteins interact. So it's a similar way we use this. You can split the DNA binding domain and the activation domain of the GAL4 protein. Okay, and you fuse each to a leucine zipper, which will dimerize. So what you have here is you now have two enhancers that express the two halves of the GAL4. Okay, and these two halves can come together to then activate transcription, right? But only where the two enhancers overlap will you reconstitute the GAL4. Okay? So this is a way to refine the expression pattern. So for example, this was the 31C06 pattern I showed you. It labels the cells we care about and probably a few others as well. And this is another enhancer that's expressed in some of the same cells. And so we were able to combine these two, and now you get a very specific expression pattern. So it's only a subset of these. Okay. Great. And so what I did was, again, inhibit these neurons and then try to look for whether this causes a change in the behavior, right? And what we found was that we could indeed block these neurons and block the increase in sleep, right? So this suggests that the output of these neurons is functionally required, right? Great. Okay, so I found these missing neurons, but the next question was really how are these neurons changed? Because a lot of the interesting things that brains do is about dynamics, right? But how circuits change in response to different kinds of inputs and so on. And so to get at that, I did a couple of experiments. One was to use to look at whether following flight disruption, these neurons get activated. And so to do that, we have a slightly old-fashioned, but actually, in our case, quite useful tool. It's called Calexa. And so what this does is it's a modified NFAT transcription factor, which is calcium sensitive. And so what happens here is that when you have more calcium, this transcription factor will enter the nucleus and express a reporter gene, which is GFP. Okay. And so it's a way to change. So of course, when neurons are more active, we get more calcium. So more neural activity, more GFP, right? So this is quite slow. It takes a few hours to reflect the changes. But in our case, that's actually very good because sleep manifests over several hours and in fact, several days in this case, right? So we're looking at a buildup of calcium over a long time scale. And what we saw was that, again, these projection neurons identified are more active, right, following flight disruption. Okay? And that's fine, but we saw one thing that puzzled me a bit was that these the sleep can increase for several days following flight disruption. So you cut the wings, but several days later you can still see increase in sleep, right? So how does that work? And so one possibility was that these neurons were structurally changed, and that because they were structurally changed, now they're able to sustain sleep for a really long period of time, right? And so to do that, to see whether that was indeed true, we turned to this tool called STAR, which lets you mark all the presynaptic sites. And that's what you see here in magenta. And so we can quantify in 3 dimensions these presynaptic sites, okay? And I don't know if the movie will work. Yeah, it does work, great. Okay, so you can see, in these different tracks, you can quantify these actual presynaptic sites, right? So this is a measure of structural plasticity of these neurons. And what we saw was indeed that there's these neurons are structurally changed. There are more presynaptic sites following flight disruption. We can disrupt flight in a couple of different ways. Okay, okay. So to summarize this part, we had discovered a kind of surprising relationship between flight and sleep. If you disrupt flight, you can increase sleep. And I had some luck during circuit dissection, so we were able to find a novel neural circuit that mediates this effect, including the modulators and so on. And the interesting thing here was that these are parts of the brain that people have not really suspected of being involved in sleep regulation before, including wing sensory neurons and so on, and neurons in the nerve cord, right? So, so the idea that the peripheral stimulation can change sleep is again something that is relatively new, but it's something that's coming up in a range of different contexts, a range of different model systems, and so on. And so that's something we're also interested in pursuing, right? And these sensory neurons we identified are actually pheromone receptors. So the idea that pheromones can change sleep is really exciting. And so that's also something that we want to follow up on a bit. And more generally, I think we like to take a kind of evolutionary approach to the study of sleep, the study of nervous systems, and at least we start with the behavior and then try to work backwards and try to figure out what are the circuits involved in the behavior and so on. And more clinically, we're in the middle of what's been called a sleep loss epidemic. That means that people are getting insufficient sleep, but there's many reasons for this. And you can imagine that if these circuits are activated or inactivated at the wrong time, this can lead to a change in, this can lead to negative consequences. Okay, so any questions about this part? I'll quickly move to talk about memory in the next part. For about, I don't know how much time I have, about 5-10 minutes, I can talk about memory. Any questions about this? Okay. Okay, thank you. All right. All right, I'll be quick. All right. So the idea that sleep promotes plasticity in learning is not very surprising, but this idea really took flight in the field after this this seminal paper by Wilson McNaughton. And so here what they did was to train a rat to run along a linear track, basically sprinkle a piece of chocolate along the track so that it'll run along this linear track. And all the time they're recording from the part of the rat's brain called the hippocampus. And so what they found was that the trajectory of the rat along this track is represented by a sequence of activation of these so-called place cells. Okay, this is color coded here. And the interesting thing was that this sequence was replayed during sleep, during non-REM sleep, particularly, right? And it's a kind of fast-forward replay. And so this paper is really influential in part because it really emphasized that sleep can play an active role in consolidating memory and so on, which was sort of known before, but people had forgotten about that a little bit. Okay, so it's all fine in rats, but in humans, sleep is particularly important for a class of memories called declarative memories. That is, memories of episodes in your life, words and facts and so on. And these are some papers that showed that. And so, but you can imagine that it's hard to get at mechanisms in humans, and it's hard to study, episodes in a fly's life or in a mouse's life, right? So what gives? So fortunately, we have this idea from Yuri Bisaki and Ed Moser that uh, that that studying in animal models is really an evolutionary precursor for declarative memories in humans. So we can study spatial, if we learn something about spatial learning in animal models, we can learn something about declarative memories in humans. Okay. So how do we study spatial learning? This is classically done in rats using the Morris water maze, which is a sort of a big tank filled with this opaque liquid, and you dump the rat in a corner, and the rat goes like trying not to drown. But fortunately for the rat, there's a little submerged platform which it has to find. Okay. Eventually it gets there, but then you have these visual cues which you can't see in this photo, but you move the cues and you move the platform, and eventually the rat goes straight there, right? So over time, you see in this classic paper from Richard Morris that the rats are learning, that they're getting faster at finding that platform, right? But if you lesion the hippocampus, they don't do so well, right? Okay, so we wanted to see if we could model this in flies. So we adapted this this kind of thermal maze for this purpose. So this is again using a thermal maze instead of a water maze. So you have a grid of Peltier tiles which are kept warm, and one of these tiles can be cooled, and then you can move the cool spot and move these visual cues, and eventually the fly figures out how to get there, right? So you can see that it's getting faster over time here as well. And the interesting thing that we found was that uh, flies show very dramatic age-dependent declines in spatial learning. So this is about 3 weeks of age. And so what we found was that they pretty much stopped learning by about 3 weeks of age. It's a very sensitive measure for age-dependent decline. 3 weeks is not that old even for a fly, right? And so, one of the things to note was that, if we tested the same kind of experiment, but using a olfactory conditioning paradigm, for example, the flies would be impaired, but not so impaired. So they'd be like there. This seems to be a little more sensitive assay for age-dependent decline. And it fits with the human literature on declarative memories as well. Okay. And the interesting thing was that these age-dependent impairments could be reversed by enhancing sleep. So we enhance sleep a couple of different ways. One is feeding this drug called gaboxetol, it's a GABA agonist. The other is using genetic tools. So both ways we enhance sleep, we can actually restore learning. So this is taking a fly that is impaired, and then you enhance the sleep for 2 days, and then it restores learning. Because this suggests that sleep can not only consolidate memory in a healthy brain, but can actually restore functioning to an impaired brain. Okay? And so this restorative aspect is really exciting. It suggests that sleep could possibly be used as a therapeutic, maybe in human disease and so on. So that's something we want to follow up on. We're working on in the lab here at Ashoka as well, trying to figure out what kinds of mechanisms are involved in this. Okay. And we see the same thing actually when we enhance dopamine signaling as well. Okay. And so that's what we're trying to figure out, what's going wrong with age, how does sleep make it better, and so on. And so I'd like to acknowledge so much of this work was done at WashU in St. Louis in Paul Shaw's lab. And Bruno and Michael helped a little bit with this, the spatial learning and so on, and gave me a lot of advice in building this assay. And so the lab is about a year and a half old at Ashoka, and those are the students in my lab, where they're young and highly motivated and doing great. And I also like to thank various people in the Indian fly and neuroscience community. Shashi and Girish were very helpful to us in keeping our flies for a year at Pune. So we're really indebted to them for that. And everybody here has been super helpful and super supportive, which has been great. Okay, so thank you. So I have to thank the funding agencies as well. Thank you.
Wonderful talk, Dr. Krishna.
Thank you.
I have two small questions. One is that you have shown the genetic changes that are happening in these Drosophila during the sleep and how they are getting reversed during with the age when the sleep is increasing. So are these genes also present in the humans for these Drosophila genes?
Are there homologs of genes present in human? The learning part, we're studying dopamine signaling. So of course, dopamine is, widely conserved molecule. So that's, one would expect that similar kinds of mechanisms play roles in, in humans as well. Um,
Okay.
And but the flight part, we had this receptor called rickets, which is uh, when you disrupt rickets, you can increase sleep and increase flight, disrupt flight, and so on. And so rickets is actually a homolog of a G protein coupled receptor in humans called LGR6, and LGR6 has also been shown to be involved in sleep. For example, there was some papers that looked at changes in gene expression following sleep loss in humans, and LGR6 was one of the most highly changed genes, and so on. So yes, we expect similar things to be
Okay,
playing roles.
and one more question I have, that you have shown that how the size of the elephant and the sleep is varying. So the quality and the quantity of the sleep, it varies across the species, right? So
Yes.
how does the brain size is affecting the duration and the quality of this?
Oh yeah, that's
Yeah,
a
your
great,
views
great
on it.
that's a great question actually. So it doesn't I don't think it has much of a relation actually. Brain size does not seem to correlate very well at all with the amount of sleep. Quality of sleep is a little difficult to tell, across species. It becomes very difficult to compare across species what the quality of sleep is. Within a species, you can compare over time or something. It becomes easier to tell whether you're sleeping lighter or deeper on a given day. But between species, that's a bit trickier. Uh, but the amount of sleep certainly does not seem to correlate at all with brain size or body size or anything. Yeah.
Thank you.
Yeah.
Thank you, sir, for enriching our gathering with a scientific discourse. Okay. And I would like to request our moderator, Dr. Angana Saikia, to felicitate our speaker with the memento. Thank you.
Thanks for the speaker, so kind of you to come over from one is coming from 60 kilometers this side and other is coming from 60 kilometers from this side. Thanks for coming and thank you.
So we will now be having a short 5 minutes tea break, which will be followed by the laboratory sessions that will last till 1:15. So I would request all the speakers, participants, and the present faculties to head for the tea. Thank you so much.